This in vitro assay demonstrates angiogenic sprouting in a high-throughput and scalable platform. This could be used to find new targets in angiogenesis. This angiogenesis assay combines a complex cellular microenvironment that includes gradients and perfusion with a platform that's compatible with high-throughput screening. Angiogenesis plays an important role in both health and disease. Understanding the mechanisms that occur during angiogenesis could help us to understand how cancers grow and how tissues repair. This method could give insight in the mechanisms during cardiovascular diseases and tissue regeneration, as in both angiogenesis plays an important role. The first time you use this microfluidic platform, you need to be aware of the low volumes and how to pipette. You can confirm the steps by looking under a microscope or flipping the plate upside down. Some steps are difficult to get a feel for when just reading the method. For example, how small the microfluidic channels are or how to hold the plate. Demonstrating these procedures will be Wendy Stam, a technician from our laboratory.
To begin, transfer the microfluidic 384 well plate to a sterile laminar flow hood. Remove the lid, and add 50 microliters of water or phosphate buffered saline to each of the 40 observation wells using a multi-channel pipette. Then, add 1.5 microliters of 4 mg per milliliter collagen 1 solution to the gel inlet of each microfluidic unit. Make sure the droplet of gel is placed in the middle of each well in order for the gel to enter the channel. After filling 5 microfluidic units, flip the plate upside down to visually inspect correct filling of the gel channels. Place the microfluidic plate in an incubator at 30